I'm pleased to welcome my colleague, partners, and friend, Stéphane Lemaire. We held the Biofoundry of Paris, a modularized Biofoundry, and we look forward to Stéphane. Yours.
No. So, thank you, everybody. We'd like to thank organizers for giving me the opportunity to present what we're doing in Paris to create the first biofoundry in France. I would like to start by basics about synthetic biology. And synthetic biology is often seen as something that aims at creating artificial systems that have potential applications, creating startups. But I would like to remind that it's not only creating new biological systems for applications, it's also a way to answer fundamental biological questions using new approaches, new concepts, high throughput, and automation. This is linked to the convergence of biology with other sciences, exact sciences, computer sciences, that promise to enable the next industrial revolution and to impact numerous industries with a major economic impact. This revolution requires synthetic biology to not only develop products and services that tackle societal challenges and industrial challenges, but also to fuel fundamental advances to engineer biology much more efficiently. We have seen a number of startups mainly that have emerged during the last years because it is possible to modify or even completely synthesize the genomes to address fundamental questions or to create new biological systems with application in many different fields. We are focused on health today, but there are applications in many other fields. In material sciences, it's possible to biofabricate some materials. In the agri-food sector, in the environmental transition, digital technologies, cosmetics, and so on. So there are many possibilities in many different industries, and of course, many applications in the field of health. I just illustrate with immunotherapy, personalized medicines, engineering of microbiomes, and these are only a few examples. So the concept of this bioproduction is to replace petroleum refineries by biorefineries. These biorefineries, they use biomass and are theoretically able to produce, should be able to produce exactly the same molecules. For some of these productions, the input in the biomass is a problem, because if you want to transition from fossil fuel to bio-based production, you need a lot of biomass. And competition for biomass is starting to increase. And for high volume application, the problem is that we use mainly bacteria and yeast for this bioproduction, and the biomass comes from agriculture. So it's in competition with our food needs. One solution would be to use organisms that, like plants, can use directly atmospheric CO2 and sunlight and can be engineered. So these organisms exist. They are microalgae, and we could use for high volume production algal biorefineries that can produce these different molecules. So I am an expert of synthetic biology or microalgae, and we started 10 years ago to think about how we could use microalgae and engineer them much more efficiently, microalgae, as we do. So we're working with Chlamydomonas reynardii. This is the major model system for algae. It's a eukaryotic unicellular alga. And like all model systems, during the last decades, we have developed many molecular biology tools. We have promoters, terminators, reporters, resistant genes, CRISPR, but all these tools were not standardized. So we were developing, each new project was new development of new tools. So the first step for us was to collaborate, make a European project and collaborate to develop the first Moclo toolkit for Chlamydomonas. And so we published this a few years ago and distributed to the community. People have started using this toolkit and expanded the bricks. So we had 120 bricks in this Moclo toolkit. We are now reaching 1,000 different parts. So we are now preparing a new version of this kit to expand the capabilities and to be able to engineer very fast and make multigenic construct extremely fast using Golden Gate cloning. If you think about the way we build in biology today, the strains are built and tested essentially manually. It's a slow and artisanal process. It's prone to human errors, to biases, and it's very expensive. So this method of construction is low throughput. And the problem is that we need to test many, many different constructs. And we need to test many constructs because our predictive models are not good. We don't know, we are not able to predict exactly the behavior of the cells when we introduce a specific molecular genetic circuits. And by contrast, when an engineer designs a bridge, does not need to build 50 different bridges to eliminate the 49 that collapse and keep only the one that is efficient. That's the way we build in biology. We build hundreds of constructs to eliminate the one that do not work to find the one that works. So we need to be more efficient in building, and for this we need to improve our predictive models. And the way to improve our predictive models is to build more, much faster. And then with the results, we may be able to build less and ultimately only build one construct, one cell, and the output will be exactly the one we need, like a civil engineer building a bridge. So if you want to do this, you need a dedicated infrastructure, which is called a biofoundry. You just heard about the biofoundry. So the aim is to make high throughput design-based test learn cycles. So you use computer and AI assisted design, and then you use robotics automation to automate the build and the test phases. And then this will allow to generate hundreds of constructs in parallel, hundreds or even thousands of strains in parallel, and many, many data that you can plug into your AI or ML-based software to try to make sense of this and improve your predictive models. As we have just seen, there are numerous biofoundries in the world. So a bit more than 30 biofoundries, mainly in the US, in the UK, and in Asia. And there's no biofoundry or no automated biofoundry in most European countries. So we and a few other colleagues decided to create the first biofoundry in France to accelerate the structuring of the bioengineering sectors and give access to different stakeholders to this high throughput engineering capacity to ensure both fundamental and applied research and to be a driver of innovation and a base for this bioeconomy through industrial partnerships, innovation programs, and creation of startups. So Paris Biofoundry is a very special biofoundry because it's a distributed biofoundry. The biofoundry is distributed on 4 different sites. The central site is in Jussieu at Sorbonne University and is the DNA foundry, which is quite similar to the genome foundry in Edinburgh. And a micro biofoundry that engineers bacteria, yeast, and microalgae. It's a unique feature of this biofoundry because most other biofoundry only engineer bacteria and yeast and mammalian cells. We have other biofoundries are located into different sites still in the region. The mammalian biofoundry located at Institut Curie, a cell-free biofoundry at Institut Michaelis in Juin-Josas, and an industrial biofoundry in Genopole. So I will take a bit of time to explain you what is it, what is doing, and what are the specificities of each site. So the Sorbonne University Foundry is organized into 4 different platforms. Fully automated DNA foundry for high throughput assembly of DNA, an automated microbe foundry for high throughput engineering and high throughput testing of microorganisms that, as I said, will engineer microalgae, bacteria, and yeast. An advanced phenotyping facility, a systems biology platform with capacity for sequencing, DNA and RNA sequencing, proteomics and metabolomics, and of course the associated bioinformatics and AI to especially for the design and learn phase of the DBTL cycle. So the DNA Foundry basically takes as input designs and outputs verified plasmids with a throughput of hundreds of plasmids per day. All the steps, assembly, transformation, colony picking, PCR, bacterial culture, extraction, plasmid mini prep, all this is fully automated, no human intervention. So we are taking off the human from the lab so that humans can focus on more intellectual tasks and not on kitchen recipes, basically. So this DNA Foundry is like a big blocks in an enclosure that is sterile with robotic arms that manipulate the samples, some cameras to monitor what's occurring inside because there's no human, but you can monitor it from outside, and a single software that manages the whole thing. If you go inside, you've got many equipment concentrated into a very small space. It's fully automated from the freezer. We have an automated freezer with more than 30,000 different samples that the robot can pick to make experiments. Of course, liquid handler, robotic arms to manipulate the plates between instruments. An hotel, which is the only place for interaction with humans. You load your samples, you load your pipette tips, you load your plates, and then the robotic arms will come and manipulate this and make all the experiments. An acoustic liquid handler, this is mandatory in a biofoundry. This one will manipulate tubes and will transfer 2.5 nanoliter droplets to decrease the cost of the experiments. And we have also a number of additional equipment, automated centrifuge, because you need to centrifuge your cultures, automated PCR equipment, plate sealers, peelers, liquid dispensers, colony picker, and automated incubators where the robotic arm can start a culture and come back the next day, pull down this culture or measure the growth during the culture. Then we have the second platform, which is the microbe foundry, which is basically takes the plasmids coming out of the DNA foundry and put this plasmid into different chassis, make the phenotype, and then outputs engineered strains that are verified and the phenotype datasets. So all the steps are also fully automated, not only engineering of the cells, but also the phenotyping with high throughput confocal images, plate readers, and anything that can be measured with images, fluorescence, absorbance, luminescence can be done with high throughput. We have, if you need more details, comprehensive phenotyping, we've got equipment for cell sorting, proteomics, metabolomics. A lot of the projects are metabolic engineering, so we need a deep understanding of the molecules that are produced by our cells. So we have a very high capacity for metabolomics and proteomics. Our know-how at Sorbonne Biofoundry includes new strategies for assembly, screening, selection, and production that are based on either sustainable production in microalgae or in vitro evolution using cell-free phage synthesis, designer strains for drug screening or synthetic organelles. These technologies have been developed by my partner Ariel Lindner here in the lab here, and Ariel will join the Sorbonne Foundry so we can aggregate our know-how on microalgae and engineering especially of bacteria for health applications. The mammalian biofoundry at Curie Institute is based on the Biophenix platform that you heard during Frank's talk a bit before, and we will expand the capacities of the foundry to which is mainly a test platform. So it has a lot of equipment. We will upgrade this equipment to improve the capacity for testing, but also new robotic equipment to engineer the mammalian cell using the DNA that is built in the foundry at Sorbonne. Currently, this platform analyzes cells that are transfected with siRNA or incubated with small molecules, and we will expand the capacity of this platform that should be fully operational in 2026. The cell-free biofoundry is the third site. It's operated by Mecalis Institute and built on the expertise of the group of Jean-Loup Follon. So this biofoundry optimizes cell-free protein production, especially using active learning. So the start is a given target protein sequence that needs to be produced and some bioproduction objectives. So you want either to maximize yield, to minimize the costs, and this drives the choice of the components for the cell-free protein synthesis. Then the range of each component in the buffer is then determined, and the automated system can be used. It drives the first set of conditions to be testing using what we call the sampler. And the sampler module allows to enter an active learning loop, which is based on machine learning models, with designer, which defines source and destination plates, so it gives instruction to the robotic platform. And then the experimenter module allows to test and monitor the yields with some plate readers, basically. And then we have the learner modules that will take this data and initiate a new round of a new plate with new conditions and with an optimization at each turn of this cycle. So this has been used, for example, to improve protein production. You've got some example here on the top, an increase by 34 using this active learning strategy of the yield of protein production, and has been also used to optimize CO2 fixation productivity for an artificial CO2 fixation pathway with a 6-fold improvement. So it's a significant asset to improve the pathway, and can be used before you implement this pathway in vivo. The fourth site is the industrial biofoundry at Genopole. So this biofoundry complements the DBTL cycle by focusing on scale-up and optimization of the processes required for industrial transfer of the technologies and the strengths to industrial partners that work with higher scales. This biofoundry has started phase 1 in April 2024 and will be operational in 2025. The industrial biofoundry will optimize fermentation, especially fermentation condition from 960 different conditions at 300 microliter, 48 conditions at 1 milliliter, 6 conditions up to 1 liter, and 1 condition at the 20 liter scale. And in phase 2, this will increase to more than 300 liters to have a capacity to test the strength not only at small scale like in the microfoundry, but also at higher scale, which is required before we can consider transfer to the industry. So if you want to go beyond, you need some partnership, and we want to work with industrial partners that have the knowledge and the know-how how to scale up to be much bigger volume above cubic meters. This biofoundry will allow to test both upstream and downstream processes, including culture condition, media optimization, target molecular extraction and purification. And this is a prerequisite before we can consider technology transfer to industrial partners. Filippo mentioned that one major problem of biofoundries is software. So software is indeed a major problem in many biofoundries. There is no custom industrial solution. No provider will give you a solution to handle all this data and monitor all your robots. So you have to develop your own custom solution or build on others. So we have been discussing a lot with an DIM building our foundry. And the team of Genopole has developed since 5 years now a digital infrastructure which is called Galaxy SynBioCAD. So it's a software that has been designed to work with the biofoundry. So it's doing pathway analysis, genetic design, retrosynthesis, metabolic modeling. So you can have this assistance for your design, and then prepare instructions for the foundry, for the robots, for the automation, and then uses active learning in order to analyze this data. This is based on Galaxy. So Galaxy is a widely used toolshed. It's like an app store, which currently has 10,000 different apps. So the Galaxy SynBioCAD is a portal that contains 39 different tools. And these tools can be aggregated into workflows. And we are now in the process of integrating with these tools, the tools from DIM BioConvS, in order to have a portal for synthetic biology and biofoundries, especially because all our software are also open source. So the idea is to aggregate the best of what has been developed in different foundries and have something that we could co-develop with the Global Biofoundry Alliance. So this is, as I mentioned, fully open source and compliant with FAIR principles. All these tools can be easily combined and connected together, and it's a very important addition. And the fact that it's driven by artificial intelligence is a major asset, and I think will be very important for the future. So all this ambition is to further expand the capabilities of this portal and to drive the operation of all the 4 biofoundries I presented that are present in Paris Biofoundry using this software, and to have to leverage the what AI can bring to biology in connection with our high capacity for engineering. So Paris Biofoundry is basically a large set of robotic instruments. It's driven by a software solution powered by artificial intelligence. So now I need to mention what we are doing, what we aim at doing is to make high throughput design-based lesson cycles. So cloning and DNA assembly could be done as a service, but it's not the major objective. We want to have design-based lesson cycles. Design and test of genetic circuits of enzymes, either natural, artificial enzyme, evolved enzymes. Metabolic engineering, there's a huge interest for many stakeholders for metabolic engineering. This includes synthetic pathway design and retrosynthesis, and also scale-up and process optimization, which is something that does not exist in many biofoundries. In parallel, we will continue developing improve our tools, new synthetic biology tools, new methods, new processes, including software tools and AI-driven engineering methods. And we will maintain a huge collection of Moklo toolkits, expand the Moklo toolkits that exist, and be a DNA repository. So it's an infrastructure that is open to academic labs, industrial partners, and startups through either collaboration and services. We are currently installing all the robotic instruments, all the machines with my team that is here, and we expect to have a progressive ramp up during 2025 and to be operational in 2025. So if you have projects that would benefit from this high throughput of the biofoundry, it's a good moment to come talk with us, because the operation will start next year, and we need to to onboard a few pilot projects to set up our instruments. I mentioned that Biofoundry aims at making some innovation, creating startups. So my lab and the lab of Ariel have already led to the creation of 3 different startups that are based on the technology developed in our group. So the first one is Biomemory that I co-founded with my partner Pierre Crozet, who is also the deputy director of Sorbonne Biofoundry. Biomemory is using DNA to store digital data. So we replacing the standard hard drive or magnetic tapes with DNA in order to decrease the carbon footprint of the digital industry. Biomemory raised up to now 23 million. Another company is Generare, which was co-founded by Vincent Libis from the lab of Ariel, that is focused on natural molecule discovery using a process that allows to combine bioinformatics, high-throughput engineering, and mass spectrometry to discover new therapeutic molecules, and they successfully raised 5 million during their first round of financing. And the third example is Bacta, which is focused on another aspect, which is materials. Bacta has developed technology for bioproduction of natural rubber using bacteria, as the name says, and has also successfully raised very recently 3.3 million. And there are many other startup projects that are pre-incubated into our foundry and many others to come. The foundry is also a host for projects. We have Flex Lab and Flex Office to accommodate outside people coming for some moments in the foundry and then discuss with people and have monitor what is doing there, and then a rotation of many startups, many academic labs. We have a space of 1,000 square meters to allow to have this facility. And I'll be happy to answer any of your questions. And if you want to engineer with much higher throughput, or you have huge, or you want to go very fast, please come talk with us. We can probably help you for your projects. Thank you for your attention. I'll show you a few videos of other foundries that already exist in the world. Soon we'll have our own video, but the instruments are not yet. But you can imagine what it's like being a foundry, even though Filippo already presented some of this. Yes?
Thanks for this interesting presentation. I have a question regarding the cells you can test, because sometimes if you work on cell line or on primary cells, for example, hematopoietic stem cell, you will not have the same amount of cells. So do you still have any threshold for each experiment and test it if it works with cell line with the other kind of
Okay.
primary cells?
So I'm a microbiologist, so probably I'm not the best person to answer this question. This should probably be discussed with the Curie foundry, which is taking care of mammalian cells. We don't engineer at all mammalian cells in my lab. So I will not be able to answer your questions about the type of cells. And as I mentioned, the Curie part, which is quite difficult for all the reasons you mentioned, will be launched a bit later in 2026. So we don't have any yet for the moment an answer to your question, what type of cells we accept or do not accept to engineer.